Bright lights. Hi, everybody. So we are so excited here at FIRST to really be able to start thinking more about research. As you know, we've been talking research for more than a decade at this meeting, and here trying to help move things forward in research, and it's really happening now. We coined the term a decade of discovery because this is the decade of discovery for our group of epidermal differentiation disorders, or ichthyosis. And what I'm going to do here with my partner Keith Choate and our teams is just try to update you on what's been happening, tell you a little bit about what we're going to do at this meeting. So our plans today are first to discuss a newly proposed classification that's based on underlying genes and their functions, and why moving to this new classification is important despite some challenges. We're going to review the science, and I was specifically asked to talk a little bit about basic science. And you know, there are more than 100 different forms of ichthyosis, but we'll just cover some of the most common ones. But I'm happy here at this meeting, if anybody has a question about a type you might have that may not be listed, happy to talk about that and fill you in. So we'll talk about basic science. I'm going to introduce you to a dream that we have that we're keeping our fingers crossed will move forward, called SHED, as a very key part of what could be the roadmap that you've been hearing about, the research moving forward in our field. And then alerting you a little bit to the productivity we've had from some of the research meetings in the past and what's planned here. So let's just start with some basic science. Now, I don't expect you to have a quiz after this or to really understand everything, but we're going to try to keep it basic to just make sure that you all understand with some of the more common forms that we're talking about why we see what we see. So the ichthyoses are all skin barrier disorders. And you know, our skin is very, very important. What does this epidermal barrier, epidermis, the outer layer, dermis, the second layer, what does the epidermal barrier do? Prevents us from drying out by limiting water loss through the outer layer of skin. And we call that transepidermal water loss, or TEWL. Different spelling of the word TEWL, but T-E-W-L, and you'll see that again. So the skin, or epidermis, protects us from all kinds of things in our environment, from what cause allergic reactions called antigens, to toxins, detergents, ultraviolet light, microorganisms, and trauma, including from our own scratching. Now, we back in 2009 proposed a last classification that includes a lot of the terms that we now use, and we thought it was time to do that again. And so over the last few years, a group that involved many of us, including 3 from the United States, worked on this new classification, and it came out in publication form in 2025. Now, why do we need a new classification? Well, first of all, we have learned so much over the last 10 to 20 years about what causes ichthyosis. So we know pretty much all the underlying genes. Yes, we're still finding a few, but pretty much most of what anybody in this room would have. We now understand the gene. We need treatments. I don't think anybody in this room would disagree with that statement. We need treatments that are effective. We need treatments that are safe. We all want there to be curative treatments, but in the meantime, we need to develop other treatments that at least can help with the scaling, the redness, the itch, the pain, all of these various factors that affect quality of life. So this new classification is based on the underlying gene and the function of the gene in skin to try to help move forward the concept of the new treatments, because by understanding, we can start to find targets that can be more specific. Now, one of the problems is that we've got so many names for these different types of ichthyosis. Some of them are not so bad because they're kind of descriptive, but there are others that are what we call eponyms. They were named after people in the past who may have described something or wrote an article, and we don't even know who these people are. And they're not even around anymore. But it doesn't tell us anything about that particular form of ichthyosis. And an example is Netherton syndrome. I would bet even those of you who have Netherton syndrome who are sitting here don't really know much about the man who was Dr. Netherton, an American dermatologist from a long time ago. Now, I do like history and I like honoring people, but we need to start moving forward and having names that actually tell us something and are going to help us in new therapies. Now, we also thought about the fact that many of these names are Latin or Greek, and we worried about them being derogatory, terms that, if you knew what they meant, could be used for teasing. And that was vulgaris. I know I met at least one person here with ichthyosis vulgaris, by far the most common type. About 10% of individuals have that, and maybe 1 in 400 have a much more serious double dose form. But the word vulgaris in Latin means common. Well, most people don't know that, but they know the word vulgar, and so that's a little bit of a problem. Hystrix means porcupine. And it's controversial, and we may kind of go a little bit backwards on this one, but we had concerns even about the term ichthyosis, because frankly, it means fish, scale. And that's a common tease also for certainly individuals who have ichthyosis. So that's something I'd love to hear your feedback on too, because we're trying to decide if that term should stay as a more umbrella term or not. We need simplified nomenclature. Now, none of us are used to using gene names, but that's because we just learned a different way. We need to be flexible in adding this as a new nomenclature. And it makes it a lot easier for doctors and scientists, because if there's a new gene that's discovered, you instantly have the new name, and it's understandable. Now, we used a classification approach that's called the dyadic approach. And it's not just for ichthyosis. It's really been across medicine that people more and more, when something relates to a gene, are putting the gene into the name. So this is a trend that's happening everywhere, and we want to be part of that. So what on earth is an ED, or an epidermal differentiation disorder? So this is really an umbrella term that's broader than ichthyosis. They're all inherited, and they're problems with how the skin matures or differentiates. So the broader umbrella terms we might have used before are things like disorder of keratinization. But keratin is something very specific, and we didn't think that was the best term. Disorders of cornification, well, that relates to the stratum corneum, I'll talk about in a minute, but really not even everything relates to that as well. So ichthyosis is part of this group of EDS, but EDS also includes the related skin types. That's kind of been a bit of an orphan name there. And also when you have scaling and thickening skin disorders that are largely limited to the hands and feet. We've subdivided this new classification based on the extent of the effect of the gene. So we have syndromic, that's SED, if it affects organs. So many of our forms of ichthyosis don't. They're just skin or skin, hair, nails, which are called the adnexal structures, kind of part of skin. But there are others where there are organs involved. And it's important to figure out early on what's going on and be able to predict and bring in other specialists to help with that other involvement. So there's SED, there's NED, which is non-syndromic, most of you, just involving the skin and the adnexal areas, maybe your sweat glands, of course, too. And then PED is palmoplantar is the word for the bottom of the foot. So if you have largely hand and foot, then you'll be classified as a PED, palmoplantar ED. And sometimes those just affect skin, and sometimes they affect internal organs as well. Yes?
So what if they have all of those?
If they're all of it, they're probably going to be syndromic. Okay? So syndromic is something that may be extensive, may have palms and soles, but it has internal involvement. And I will say that, to me, there's a little bit of a problem in definition because there's such a spectrum with some of the SEDs, right? Okay, so these are then further grouped by function. Why? Because if we know what the gene product, the protein that comes from the gene does, that helps us to think about new directions for treatment. This is just to show you somebody used AI to put together epidermal differentiation disorders throwing the umbrella. By the way, there are a lot of errors here. If you know anything about AI, sometimes they hallucinate. And if you're sharp and you know about all of these, you can catch at least 3 errors. Getting them fixed. So how does it work? So let's go to the most common one, ichthyosis vulgaris. So there's a gene descriptor and there's a phenotypic descriptor. The phenotype is what we see. It's what we see clinically. So the gene descriptor is filaggrin. And that is the gene itself. And then it is a non-syndromic disorder. So it has a simple name of filaggrin-Ned. Now, I'm not sure that's easier than ichthyosis vulgaris, but it's not any harder. And it doesn't have terms that we don't like. Now, Netherton syndrome, the only gene is SPINK5. That is classified as a syndromic because there can be internal involvement of different types with that. So it's called SPINK5 SED. And we do this with all of the non-syndromics. We do this with all of the syndromics. We do this with all of the PEDs, the plumoplantar ones. I'm just showing you some examples here. Now, I would say, I went through the list of people coming, and maybe there were more added in the last month, but you can look up here and you can spot your ed. But I do want to give a shout out to Denise. Where are you, Denise? Are you in here? I can't even see. But Denise has put together a spinning wheel. And make sure that you look at the exhibits out there and spin it and find your gene. So what's your name? What's your gene? What's the new name? So make sure you take a minute to look at that today. So how does my form of ed affect my skin barrier? Well, let's go back to what is the function of gene products in the epidermal barrier. So I mentioned before the stratum corneum, sometimes called the outer cornified layer. And the stratum corneum we think about as bricks and mortar, and I'm sure you've seen all of these kinds of descriptors, but just to remind you that when we get into the stratum corneum, our skin cells are called corneocytes. They are keratinocytes, which make up the cells in the epidermis. And then they flatten, they tighten when they become corneocytes. And these are like the bricks. And they're surrounded then by a dense cornified envelope of very tightly cross-linked proteins that help with this barrier. And that's called the cornified envelope. The major form, the major protein in keratinocytes is called keratin. And many of you have heard the word keratin because you have a keratin gene change. Now, keratin forms a network of filaments that aggregate with granules in the stratum granulosum, the next layer down. And you can see these granules as they're forming in there. And you can see they're still there. That black is still there in the outer layer. So that granule content aggregates with keratin. And then the mortar is what's in between, and that's fats, that's lipids that are in between, and that's also very important for the barrier. A little bit more about the stratum corneum. So at the border of the stratum granulosum to the stratum corneum is where these keratinocytes become flattened, and they lose their central nuclei. They become corneocytes. And then that cell membrane becomes the cornified envelope. And in between the cells, there are what are called desmosomes that are also important in ichthyosis of certain types. Now, it is those corneocytes, the bricks, and then the intercellular lipids that form the stratum corneum barrier. And it's that tight cross-linking that's very important in giving the mechanical stability of the barrier. But then there are what are called omega hydroxyceramides. Those are just some of the ceramides, but they are very important in being in between, in linking to the cornified envelope, in becoming a scaffold for other lipids then to form that lipid intercellular component. So let me say a little bit more about lipids. They are something called lamellar bodies, and they contain some early lipids, enzymes, and other proteins in them. What you see here with red showing the lamellar bodies is an electron micrograph. We don't do those too much anymore. We used to do a lot of electron microscopy. And you can see these things. And so that's a lamellar body. And the lamellar body contents are extruded into this space where the stratum corneum starts, and then they become that lipid in between. Now, I mention this in particular because the extrusion can only happen if you have something called ABCA12 transporter. Many people in this room have a problem with ABCA12, and that's what's happening. You can't get the lipids out and into where they're supposed to be in that outer area. And there's about a 1-to-1-to-1 ratio, a little bit more ceramide, cholesterol, a little bit less free fatty acid that is in that mixture. And then they're very tightly ordered in there. They have to be tightly ordered in order to be a barrier. So the ceramides align closest to the cells. They build off that omega hydroxyceramides. And you've got the cholesterol that binds to the ceramides, and then these very highly organized sheets. So here's a fun fact for you. I think everybody may have heard of ceramides. They're all over, they're in products, et cetera. And they're a real problem for many with ichthyosis. So fun fact, do you know how many unique ceramides there are in stratum corneum? 5, 6, 12, 20, anybody have any ideas? Hundreds and hundreds of unique different ceramides, and that's one of the problems. There's so many, and we're just learning now about what their function is. So depending on how you look at them, you may have about 300 come up. In our untargeted work, we have almost 700 different unique ceramides. If you do laborious multi-step work, you can get up to 1,300. Now, most EDS in what was formerly called autosomal recessive congenital ichthyosis, a very broad term because so many forms of ichthyoses are recessive and so many are present at birth, so it didn't really tell us too much. But many with that grouping are actually within what is now the ceramide synthesis and transport pathway, which is shown here. And if you look, I put in black some of the more common genes that we know can cause ichthyosis, ALOX12B, ABCA12, ichthyon, also called Nepal4, and you might spot some of the other ones there that you might recognize. And I mention these because every one of these makes an enzyme that's critical to get these ceramides made, especially these ultra-long ceramides that we're talking about. So we need to understand this better. And you might see towards the right transglutaminase 1, or TGM1, which is also very important in the cornified envelope and then the cross-linking. So we need to have all of these in order to have not just the ceramides, but the ceramides attached to the cornified envelope. Now, this is the stratum granulosum, and here, as I mentioned earlier, we have keratinocytes filled with granules, largely made of prophylagrin and some keratin. And the prophylagrin is broken down to form phylagrin. And you've heard that term from me before, because phylagrin's a problem when we have what's been called ichthyosis vulgaris. And then these bundle with the keratins to form in the stratum corneum part of that very important barrier. Transglutaminase 1, or TGM1, is actually a problem in the most common form of congenital ichthyosis, what we've called lamellar ichthyosis most often. And so the phylagrin promotes cell flattening, and it enables the cross-linking by this enzyme called transglutaminase 1, which is critical for forming a barrier that is insoluble. You put water on it, it doesn't melt. Phylagrin also breaks down, as very important for us, into other substances that help us to acidify our skin. And you'll hear why that's important in a minute. Now, 1 in 10 individuals has a deficiency that's genetic in phylagrin. And that means within this room, there are many people who have that. And I mention it because we're now kind of interested in modifiers. So you may have a certain kind of ichthyosis that's made worse because you also have a deficiency of phylagrin. And this is something we're starting to look at, and you might be hearing more about modifiers like that. But that's the first one that was discovered many years ago. I just want to mention, too, the process of desquamation. Desquamation is scaling, is skin coming off. and the role of pH. Now, you know pH, that's how acid or base. You got something like water, usually about 7. Our skin is a bit more acidic, especially at its surface, and that's really important for normal desquamation. Now, desquamation is regulated by a whole bunch of proteins called kallikrines. They're breaking things down. If there are too many of them, they're breaking down proteins, they're causing inflammation, and they're inhibited by an inhibitor called lectin. The I is for inhibitor, and that is what's made by SPINK5. So those of you who have Netherton syndrome, you are deficient in this, and that means you have a lot of kallikrines, your barrier breaks down more readily, and you tend to be more inflamed. And there's an example of that, showing the desquamation that can be part of that. Now, we talked before about lamellar bodies. Everything kind of comes together. Lamellar bodies is where these kallikrines and the lectin are stored and secreted with the lipids into that intercellular space. So you also need to have ABCA12 protein for that. Now, here's some examples of some of the more common forms. And so we have filaggrin NED, ichthyosis vulgaris, insufficient filaggrin. We've got the next most common form, which was called X-linked ichthyosis, only occurs in boys, due to a steroid sulfatase deficiency. STS is the gene. And here you've got problems with dissolving desmosomes and shedding skin. Here's an example, the most common form of the keratinopathic ichthyosis, keratin 10 NED epidermolytic. And we're going to start shortening some of these terms to make them easier. But here we have, if you look at the picture on the right, that is what it looks like in the skin. And the first layer of skin cells is fine, and everything after that, because it very much relies on keratin 10 or its partner keratin 1, just busts apart. It lyses. And as you can see, it kind of looks like there are a lot of spaces in there. And then that pink stuff overlying it is the stratum corneum, really, really thickened. So you have cells that are kind of falling apart from each other. And the response is to create this compensatory thickening of the outer layer. This is a picture of the SPINK5 SED with the absence of Letke and very high kallikrines. This is transglutaminase 1 NED, or a lamellar ichthyosis, problems with cross-linking to form the barrier. And then we have a patient with very red skin. This could be harlequin ichthyosis, which is always ABCA12 NED, or a form of what's been called CIE, which is also a large group, congenital ichthyosiform erythroderma, with many, many different genes, but they're in that pathway. Functionally, they're all in one group because they help us to make those ceramides. Okay, and a little bit about the group function before I go on to a few other things quickly. This is why we want to be grouping. So the keratinopathic disorders, this is from one of the publications that was put out by the group with describing the new classification, the keratinopathies, and there are, of course, several different keratins involved in ichthyosis. Keratinopathies are not just ichthyosis. There's a blistering disease that's due to keratinopathies. There's a PED, a palmoplantar epidermal differentiation disorder that's due to keratinopathies. And there's progress going on in those areas with clinical trials. And I think some of those clinical trials, if successful, could also be used for keratinopathies that lead to ichthyosis, because the mechanisms are very similar in all of these. So grouping these keratinopathies and thinking about them as keratinopathies will help us in certain directions. And I just listed here some directions that we think are actually very interesting that are moving forward, related to the specific type of inflammation, related to the ability to selectively ubiquitinate. And I doubt you know that word. I barely know that word, but it basically means that you are clearing, all of our proteins get cleared. So if you can selectively clear the abnormal keratin, that would be an interesting way. And that's actually being done right now in another keratinopathy. And then possibly the fact that we have found increases in a particular pathway that leads to overgrowth of the epidermis. And then we talked about all of those different ceramide synthesis and transport disorders. Wouldn't it be great to be able to somehow provide an end product that would bypass many of these and maybe even prevent the buildup of toxic ones, which I think can also be a problem. So I'm going to end this section with just mentioning challenges. We're used to what we know. We don't want to learn new things. You're born, you're given a diagnosis, it's what you're looking for. That's going to be hard. We know that. And it's going to take a while. This is not going to happen overnight. But we think that that's the right direction to go. Access to molecular diagnoses, if you don't know what subtype you have, we're lucky here in the United States, we have the registry, and if any of you have not signed up for the registry, you can find out what gene you have by being part of that. So please, please do that at this meeting. We know that these terms are part of groups. First, PC project, decoding DREAs. And so we need to think about that. And like I said, we may keep the term ichthyosis. We're starting to think about that a little bit. One of the big problems for me is that it takes everything to a gene level. So when you think about what we used to call CIE, with 10 different genes or possibly more, well, we don't have a term yet for that. But we just the other day were talking about just substituting it with CST, for example, ceramide synthesis and transport, because that's really what it is. And we need some better definitions. What is truly syndromic, what is not? But there are advantages, and I hope I've conveyed some of those to you, being able to have comprehensive classification. The articles that came out with this are a beautiful compendium that's a reference for doctors, and I'll show that to you in a minute, so have your phone ready. Better characterization of patients means better clinical trials. Ultimately, gene-based names may be easier to remember, and understanding pathogenesis improves the chances of finding new therapies. So here's where, take out your phone if it's helpful for you, because how am I going to remember these? Well, there are 4 articles that are a reference for this. And usually once articles are out for more than a year, which they now have been, they're available in the public domain. And if not, we'll make sure that FIRST gets it posted on the website. But these 4 articles have every single gene in them. And if your doctor does not know about them, you can provide a copy. OK, I'm just going to quickly run through a few other things. We have a roadmap here for FIRST. It's really a wonderful roadmap that is very ambitious, will take a lot of money, and we're fundraising, and we're really going to be behind trying to do everything we can ultimately to get new ways to treat ichthyosis. But this was an opportunity that Keith Choate and I seized upon, and our fingers are crossed that this may actually move forward. The National Institutes of Health has a rare diseases clinical research network. There's not been anything involving skin. But it allows individuals who are part of this network, groups that are part of this network, I should say, to facilitate clinical research, and it shares very much in the cost of that research. It helps to establish infrastructure, establishing uniformity in studies so that there are parallels. Engaging patients and advocates is a requirement, so that's wonderful, and helps new investigators in rare disease research. Well, we know we have a high unmet need here. We've had paradigm-shifting technology. Many experts, clinicians, scientists in the United States, but not really coordination, except through maybe our MSAB here at FIRST. Well-established advocacy groups, including FIRST, who can partner, and then a growing interest. And I'm really excited about the growing interest. Scientists, clinicians, pharma, the FDA, the NIH. And so we did put in an application for something called the Consortium for Hereditary Epidermal Differentiation Disorders, or SHED. And SHED, if funded, would be the first rare skin disease in the RDCRN. And it would also help us to jumpstart the FIRST roadmap. Don't know if it's going to happen. It looks like got a good score. It looks like it's going towards approval. But unfortunately, the NIH then has to make funding decisions. And so we'll see. It's not an inexpensive venture. But if we have this, we'll get bringing together the complementary expertise, not just of people who know anything about ichthyosis, but also we're partnering with many other people who can bring in other expertise we don't have now. We really need to increase the pipeline of clinical and research experts, and it'll help. We need to understand better, with your help, the unmet needs and reassessing these needs as they evolve. We have been working hard to establish what's called clinical trials readiness so that our clinical trials can move forward with industry partners and others and have meaningful outcomes. We're trying to evaluate the natural history of different forms of ichthyosis. We don't even understand that. We did a double-blind trial recently, and it was clear that off of any kind of medication on placebo, people were flaring, they were getting better, flaring, getting better. We need to understand that better, and we need to understand what we call comorbidities, what you can have with an increased risk because you have ichthyosis. We want to improve diagnosis and risk predictions and explore how what your gene tells us translates into what we see as part of your clinical picture and understand the pathways between the gene and what we see clinically. And finally, the new directions in therapy, very important. So just quickly, we're organizing this as an administrative core that brings in a lot of other talent, working very closely with FIRST, and PC Project was the other advocacy group we brought in, and then a large group of investigators from all across the country who've been engaged in doing this, including at what would be 7 initial research sites. But that's not all, because we have genetic teams at each site. We organized an additional 37 referring dermatologists from 32 institutions who would be part of this network and sending us information back and forth, communication, really spreading information and helping us to move forward. And then, perhaps most important, the Patient Advisory Council. Some of you are involved. We capped it at 20 because you can't have a group that's too big. But we did enlist a lot of people. Everybody said yes, who would be representatives, giving us feedback, providing input. And then a lot of resources at the NIH that will help with how we move forward logistically, communication, oversight, and tracking output. So I'm just going to briefly mention 3 projects. You're going to hear a little bit more about how these might move forward. One is natural history. I'm just showing you all these things. This is what a clinical trial list of everything we would do would look like. I hope you can read that. Standardized photography, I want to spend a minute just mentioning that. You're going to hear about that in a minute. You're going to hear how Keith Choate and his group has moved that forward. We have been doing standardized photography using his technology now for a very long time, and it will be part of it. So we have complete body pictures on everyone. We have already developed this new ichthyosis severity score, and it's been validated fully, and now it's been automated. You're going to hear about that in a minute. At this meeting, we will finish the PRED score, and I'm going to talk about that in a little bit as well. And I just do want to mention that through your help at other meetings, we have further added to a specific quality of life score. There's what's called the iQual 32. We've now added to that. And then using machine learning as part of this so we can query electronic health records and help with that. So some of the things we're looking at are age-related changes, growth and development, severity fluctuations, spontaneous improvement, and then what other problems exist with ichthyosis. We're going to be able to engage neonatologists for looking at prenatal and postnatal complications in a way we couldn't do before. We have a second project that is for those with keratinopathy. That's just the first. But we had to start with something, and we chose this, and we got an industry partner to just give us the drug for free. So we're going to be looking at a project that's based on some work that we've done in our lab showing that this pathway involving epidermal growth factor is increased. We start with an early phase trial here where we dose escalate, see what happens. You can see the list of everything we're going to be capturing in this group, and that we're going to have observation first, followed by the dose escalation, and then an open label period afterwards to be able to continue. So if we can move forward, and some of you have a keratinopathy and we can get you to one of our clinical sites, we'd love to have you involved. And it's not just looking to see what happens. We're doing some real basic science, looking at the levels of the drug in skin, for example, and of course, looking at biomarkers of response, and that's the last big project here. We're going to be doing 2 studies, that and also systemic retinoids. Many of you are on systemic retinoids, but you know there's never been any real study for the last decades of that to understand what's happening in your skin when you're being treated with a retinoid orally, and I could say the same thing for possibly if you're using it topically. So this is work that will be spearheaded by Keith Choate, analyzing skin and blood biomarkers in a multi-omic approach. So we're not just looking at genes or RNA or proteins or lipids or microbiome, but doing all of them and then combining them on you. And then analyzing these various signatures, trying to understand them, pulling in AI, and comparing these profiles to try to ultimately figure out candidates for targeting. Just a little bit of a heads up. This is some of the work we did in past meetings, where we used some of the earlier techniques we had to look at the RNA. And you can see, what I want you just to notice, different patterns. So depending on what subtype of ichthyosis you have, the patterns, the colors of the red that goes up and the blue that goes down look different. This is just an example from the tape strip lipidomics that we've been doing, showing specifically not reductions in ceramide, but increases in ceramide. There's so many reductions, it's hard to put in one heat map. But you can see, again, if we're looking at transepidermal water loss and the severity of scaling overall and then redness, that there are different patterns here, again, among the 4 types. Why is that? Same thing for microbiome. And then you're going to hear a bit about these in a minute. The registry with genomics, transcriptomics, part of the biopsy, some very special advanced transcriptomics, and proteomics with stamps. So stay tuned. But I'm up here telling you about research that can only happen with your help. So if there are some of you who want to be involved in research, you're going to hear this from Keith in a minute too. There's so much that you can do. We need normal skin controls as well as individuals with ichthyosis. And it's not just a biopsy. If you don't want to do a biopsy, we've got a lot of other things that you can do that are what we call non-invasive. So please sign up. Every sign up is in the registration area. Get involved, and if nothing else, we'll be asking you to do some surveys. Are you in the registry? And if you're not, raise your hand if you're not in the registry. I know this might be embarrassing, but I'm just wanting to show there are lots of people who aren't in the registry. You should be. We also, clinical trials readiness. We have a device that's been invented by one of the top sensor engineers in the world that will measure tool. You can take it home. Lots of things you can do. We want to be testing it here at this meeting. So I don't want to run out of time. I want to give plenty to Keith and the group in a minute. But just I'm going to quickly show you. There's a lot we've learned. This came out of the registry. This is an analysis of preterm birth rates. And it gives us information about who is being born prematurely, who's not being born prematurely. And some actually have one group, the STS one, which we kind of understand, has a reduced risk of prematurity. But really, we now want to know why. So that'll be something we're thinking about. The bullying studies that some of you have been involved in, that came out in 2005. Really important to ask about bullying when you're seeing your doctor, particularly for children, but it happens with adults as well. And another example that we just sent back the revision on for the top allergy journal is that we've discovered the comorbidity of allergy across every single group. We captured that at the last first meeting. This is a little bit more on the ISS, the ichthyosis scoring system that's been validated now. And we were able to show something that's very important to show, that it's not just in light-colored skin, but it actually is valid in darker skin types as well. We've been developing other tools here, including what I mentioned before, adding to the quality of life measure that's been validated, because we found that there weren't enough questions about how much time it takes. What's the financial burden? And so we added a few. That was tested and validated here as well. And the patient-reported ED score being completed at this meeting. We've never had a way for you at home to self-assess in a validated way. I just want to let you know that tape strips showed the same thing as doing a biopsy when we looked at markers using RNA analysis. And we've done more work thinking about biologics. I'm not going to spend any more time on this, but just letting you know, some biologics work for some people, some work for others. We really haven't been able to figure out. We know certain types we wouldn't use it in now. Really more the more red skin types is what we predominantly will use these in. But that's one of the reasons it's so important to be doing some of the research, to be able to predict in advance based on your immune type, whether you might respond to this biologic or that biologic. And I do want to just mention about itch. You may have even started or tried a drug called dupilumab. I don't know how much it does with ichthyosis for the skin manifestations, but it has really been helpful for several people who have itch. We have a study going right now that we largely did in people who have genetic blistering diseases, but we put in some patients, particularly with the keratinopathies. And this is just showing you we had an observations part. We treated for 16 weeks. There's a 5-year long-term extension. They had to have bad itch. And we really did show clinically meaningful reductions in worse than average itch, some reductions in pain, improvement in sleep, but no change in anybody in the appearance of the ichthyosis, just in some things that do affect the quality of life. And we've tried this now in a few other people outside of the study and are publishing some of these as well. Finally, lots going on with SPINK5 SED. Some of you may have been involved in a trial of an anti-interleukin 36 biologic. Didn't work. That's why you have to do these trials. It didn't work. And I will give a shout out also for some of the spatial transcriptomics that's coming up now, because it's really important to try to understand all of this. There is recombinant LETK, that inhibitor I told you about, available, coming out in a harmless bacteria, and we've got a representative here who could talk with you about that. Inhibition of kallikreins by a topical inhibitor. We've got a representative here on that, and also a systemic anti-kallikrein 5 inhibitor, also representatives here. So if you have Netherton syndrome, you got a gold mine of people to talk about potential trials. And then what you heard before, we are publishing guidelines. They're all done. We're just waiting to hear from journals about getting this out there in the public domain, but no more will a neonatologist or a dermatologist not familiar with ichthyosis have to struggle with a neonate, because this consensus report comments on all of these aspects of neonatal care with detailed instructions about what to do and what to think about. And it's really going to become the go-to reference for babies with EDS globally. And I want to thank Pedra and FIRST for funding that work. Now, I'm going to stop and pass this clicker over to Keith. Keith, are you ready? Who's going to talk a bit about, I think we'll hold questions for the end because we're running a little bit behind, but Keith is going to talk about some of the research that's being done at this meeting.
So I'll say that Amy Paller is the only person I've pulled an all-nighter with since college, and not just 4 in a row the first time we submitted this CHED grant, and again subsequently. And she and I have traveled together at every meeting and found spaces to work to try to get CHED to fly, and I think it really will. I wanted to just readdress with all of you the importance of being in the registry. In fact, all of the work that we've done over the last 10 meetings where people have enrolled really created the registry, and that's the foundation for having CHED in the first place. And so we have been incredibly grateful to all of you for participating. I also want to point out that there are 15 physicians who are here who are doing clinical screenings. There are another 20 people between all of our groups from the Paller Lab, from the Choate Lab, and volunteers that have joined us that are helping us to run really a pop-up clinic today where we're going to see over 80 families. It is really important that we have all of you participate in research, and it is flexible in terms of what you do. So even if you're already in the registry, you can still do tape strips. Even if you have already done another piece of another project, please come do surveys. And if for whatever reason you knew you were doing research today but you forgot the time of your visit, please go to meeting planner B to talk with Angela, who can tell you about it. And if you missed your meeting, your visit with us today, because you were so engaged in everything else that was happening, that's totally okay. We can find ways to work you into the schedule, and we can accommodate anyone who wants to be a part of research over the course of the weekend. So let's talk for a moment about the registry. So, ultimately, I have been a strong believer that genetics is an incredible tool to understand how EDDs arise and how to develop better therapies for these. And so we really want to develop new approaches for therapeutic intervention. And now we have tools that we've never had before that allow us to pair that genetic data with systems-level understanding that comes along with comprehensive phenotyping. Those are the photos that we take. Those are the surveys that we're doing. Blood and tissue multi-omics, we talked about needing to understand. We think about DNA changes, but we need to have an understanding of how that changes RNA expression, but even RNA expression at the level of an individual type of cell that sits within the skin. We need to understand the proteins that are secreted by those cells, the proteins that are present inside of those cells, the lipids that are a part of the tissue, and then also how the bacteria that lives on the surface of all of our skin is interfacing with these cells to ultimately generate what we see in terms of our skin biology. And we also mentioned before this idea that by engaging in therapeutic trials, we can also better understand why drugs work. And so this idea of having a biomarker of severity is one of the most important things that we can find to be able to do better clinical trials. So registries really can enable research. Since we started, we've enrolled 1,725 kindreds. We've been able to provide genetic diagnoses for free to 1,450 families in the United States and around the world. And many of you have been a part of this, but we simply get a history, we do some photography, and we collect saliva DNA. To be able to move forward. One of the things I do want to say is that if you already have a genetic diagnosis, that's only part of the answer. We actually still need your saliva, because that idea that we were talking about before, that other genes may ultimately modify the way that ichthyosis appears or manifests in an individual, that wasn't done when you had your genetic testing. So even if you had genetic testing before, we're going to take it to the next level. So please come provide us with another sample so that we can do that important work. We know that there are additional genes for EDDs. Some of you who are here at the meeting, a minority of you, thankfully, about 10% of people ultimately don't have a genetic diagnosis, and our group has identified 12 novel genetic causes of EDDs over the last 15 years, and I think that we're going to be able to continue to find more. And every time we find a new gene, it unlocks a new piece of biology that's relevant to scaling and redness and helps us to move closer to developing more effective treatment. The registry has also been really central to enabling us to facilitate research broadly, both nationally and internationally. So registry data has supported the development of severity assessment tools like the ISS that we co-developed with Dr. Paller. It's provided new information about clinical features, so we were able to show the first data on how ichthyosis affects mental health, early life growth and development, eye, hair, and nail complications that have led to a number of publications. We've been able to enroll multiple clinical trials. So all these people who are coming out with new therapies initially say, we need individuals who have a specific genetic diagnosis that live a certain distance from one of our sites. We're able to instantly give them that information. And actually, we don't give it to them, we give it to you. Because you're a part of the registry, we reach out to you and say, this company is doing a trial that's relevant to your disease type, to contact them, proceed in the following way. And so we're really seeking to nucleate connections with clinical trials. We also have, over the course of all of these meetings, the biopsies that we've been able to generate have produced cell lines. And these cell lines are being used by investigators who are understanding fundamental biology of lipid biosynthesis and transport. They're being used to develop better approaches to delivering gene therapies. And they're being used to do preclinical modeling of potential new therapeutics. And so I think it's important that the registry serves as a foundation for the research that we do. And so the question that we need to ask ourselves is, how can we answer meaningful questions? And I think Dr. Paller touched on this, so I'll breeze through it. But we really need to understand individuals' experience over the course of the lifespan. A lot of people have questions about what's going to happen when I'm 70, 80, or 90 years old, and I have a specific type of NED. We need to really understand comorbidities more granularly than we do today. What are the other things that are associated with the disease? How does skin burden change with age, season, or location? So many of my patients have told me that they have a skin cycle. We need to understand that cycle and what the drivers of it are, both in terms of how it looks, but also molecularly by using other techniques. We want to understand what are the common biologic pathways that drive inflammation, scaling, and itch, because ultimately we might not need to have bespoke treatments for each of these EDDs, but in identifying common pathways, be able to develop individual therapeutics that can treat multiple different genetic causes of disease. I think we're starting to define the clinical data elements, patient-reported outcomes, and biological samples we need to answer these questions, and the SHED will allow us to broadly share them. And so one of the things that I'm most excited about is the fact that we have a whole new generation of researchers that are rising up who are going to one day replace Dr. Paller and I here at the podium. And so I'd like them to share with us the research that they have. And the first presenter will be Dr. Agazadeh. Is she here to give her presentation? All right, so we're going to have Dr. Paller come on back.
You can hear me,
Is
right?
your lavalier on?
I got it on. All right, so Dr. Agazadeh, who is doing research with us today, is not in this room, I'm so glad she's now part of FIRST because she's at the Mayo Clinic, not far from here, but is very interested in women's health. She's a pediatric dermatologist who's an expert on genetic disorders, but she's very interested in women's health. And we haven't really had an expert on that here before, so we do now. So as you can see, she wants to involve, a lot of people in this room are female, that's good. We need to understand a bit more about vulvovaginal symptoms. We need to understand more about female sexual function. There's been a request to FIRST to do research in this area, and we have an opportunity now. So this is just a survey, but she wants to hear from you, and there's going to be some time at this meeting, too, to have some discussion about that with her. So please think about that. I think you can be on the lookout for this if you're female. I mentioned before PRED, and we are doing our final testing of PRED with you, including, as you'll hear, with photography. So we're going to use the photography to validate questions that you may be answering about redness scaling. But we're also going to have questions in there to validate more about itch and quality of life. We heard from FIRST that a gap in what we've asked you about has to do with pain. Not everybody has pain, we know that. But we really haven't studied pain in ichthyosis. So we have a survey here at this meeting. On your tables, there is a QR code. Just put that on your phone, and when you have a few minutes at this meeting, you can answer questions. The first question is, do you experience pain? I don't care whether you do or not. Please sign up for this. And if you don't experience pain, say no, because it's also important for us to understand how much of a problem this is, and if we can link it to certain subtypes, et cetera. So we would like more than 100 people, or parents can do this as well, to respond. And so just to remind you, these are some of the questions with PRED, and at this meeting, we're comparing about that, and this is the pain survey. It's not very long, and hopefully you can answer these. So we'll call up now William. Yeah, there we go.
So William is William's a Yale undergraduate who's been working with us on developing ways to better assess ichthyosis severity. One of the things we mentioned is how important it is for us to understand severity over time. And so when you come to one of our clinics, we'll take photography to do this, but we want to be able to put this tool in your own hands so that you can take a piece of information and take just a couple of photos and instantly read out a severity score. And so so William's going to tell you about the foundation for that.
Yeah, and thank you, Keith. So what we've already developed, again, some of these tools are things that we've already taken, like the first couple of miles, but they're not quite there yet for something that you guys can use at home or with your own phone, for example. And so the goal for the future is what we already have today and where we want to go in the future with this tool. What we already have today is a tool where we can take a standard clinical photo and we can instantly generate a severity score based on the ISS for this photo. And the utility of this is here's some of the results that we've had so far. We've compared it against results from 14 different dermatologists. Each one rated these standard clinical photos, and we compared it against the performance of our model, and it's very consistent with the model so far. However, right now only works for standard clinical photos. What we would like to do going forward is to have a tool that you can use on your smartphone. You don't have to come in with, a professional photographer or in the clinic. You take these photos and you can assess the quality of your skin and like the severity of your ichthyosis directly at home. What that can tell you, for example, is this getting worse, for example, in December versus August? The doctor just prescribed, new treatments. It's been 3 weeks. Has it gotten better or worse over that time period? This is me, where I'm living this summer, with my own phone, and what I'm already able to do is be able to capture even a relatively messy background, the silhouette of my body, as well as a projection of like the 3D construction of my body. And so we can assess now the surface area of the skin that's been involved. Is, for example, my hand worse than my torso? What we don't have, however, is how do we take this tool and put it in a smartphone that you guys can use to assess the severity of your ichthyosis based on the validated measures that we've already done. So what we'd like to do is, one, tracking over time, right? Having the resources to have a professional photographer or come into the clinic with the expertise of a dermatologist is not accessible year-round. And so if this is something that you can do with the phone in your pocket, that gives you more data for what is the treatment that we've been talking about working. If we want to look for new treatments, right, how do we assess the longitudinal effect of whatever the treatment is over time? That's not a time point that we have right now. And finally, we would like something that is precise, but also accessible to each person in this room. Having access to a research center sometimes can be a privilege that not everyone has. And so if this is something that anybody can download and use on their phone, one that gives us more data to improve this going into the future, but it's also something that maybe you don't have to fly across the country to see Dr. Paller or Keith or someone else who spent years upon years looking into these diseases, to have some idea of how severe your ichthyosis is and what can we do to make it better. So what we're asking today is, we've already shown that this model works at the expert level on professional photographs. Today in the photography room, we have our photographer again, but in the next room, we have just an iPhone like mine set up. It's secure, it's not directly my phone, but what we'd like to do is pair the professional photographs with iPhone photography. We'd like to see if we can have the same quality and standard with just your iPhone, taking these same photos and assessing in the exact same way. This is important not just for people with ichthyosis, but also the family members. We want to have a baseline, an unaffected standard to see whether this model works not just for affected patients, but also like unaffected people. And so family members will also be very welcome. But thank you again.
If you haven't enrolled to do this really exciting study, please do so. Our next speaker is Angela Lowe, who's going to present additional projects.
Hi everyone, my name is Angela Lowe. I'm a fourth-year medical student at Yale. A lot of you probably already know me from phone calls, emails, since I'm the ichthyosis registry coordinator. So like Dr. Choate said, I'm going to be presenting on 2 projects that have resulted from your contributions to our ichthyosis registry. Oh, and how do I go back? So the first project that I'm going to be talking about is growth patterns in children with epidermal differentiation disorders, or ichthyosis. So we conducted a longitudinal registry cohort study from individuals enrolled in our registry. We obtained growth charts from 146 children with EDDs and 27 unaffected siblings. we compared height and weight data with CDC standards, and we did that between the ages of 0 and 18 years old. And the following genotype groups had enough sample size in order to do genotype stratified analysis. So ABCA12 NED, or Harlequin ichthyosis, and congenital ichthyosiform erythroderma, keratin 10 NED, or epidermolytic ichthyosis, FLG NED, or ichthyosis vulgaris, STS SED, or X-linked ichthyosis, and then SPINK5 SED, or Netherton syndrome. So what we found was overall for EDDs, height and weight were significantly below the 50th percentile in early ages, but were no longer statistically significantly different from the 50th percentile after 48 months. We also saw that genotype strongly influenced growth outcomes, with ABCA12 and SPINK5 showing the most pronounced early life reductions in height and weight, while filaggrin variants remained closest to the 50th percentile reference standard. And STS mirrored the reference standard throughout all ages. All genotypes with early growth deficits showed growth recovery to the 50th percentile and beyond the 48-month mark, except SPINK5 SED, which showed persistent height deficits beyond 48 months. When we compared affected individuals with their unaffected siblings to control for possible environmental factors affecting growth, we found that affected individuals had mean weight and height percentiles significantly below the 50th percentile early on, whereas siblings were always at or above the 50th percentile throughout all ages. We proposed that changes in body surface area dynamics may contribute to the growth recovery. Infants have a higher surface area to mass ratio, leading to greater evaporative water losses through the skin for a given degree of barrier dysfunction. And as that ratio decreases with age, this could explain the early growth deficits and later normalization that was observed in many children in our EDD cohort. I want to emphasize that your participation matters. By enrolling in the registry, we were able to solve a lot of your genetic diagnoses. We were able to contact you about this exciting opportunity, obtain your growth charts, and do this wonderful project. And then your data is really what allows us to provide that anticipatory guidance and provide genotype stratified guidance and change what we expect to see for different individuals with EDDs. So you're really helping the next generation of individuals with EDDs, and we're really grateful so far for everything that you've given us. So the second project I want to talk about is validation of the ichthyosis scoring system in individuals with epidermal differentiation disorders. For those of you who don't know what ectropion is when the eyelid, most commonly the lower eyelid, falls outward and away from the eye. And in individuals with EDDs, this is typically caused by mechanical traction from the scaling around the eye. And consequences of ectropion include an inability to close the eye or logophthalmos, eye reddening. This can also cause blurry vision, light sensitivity, dry eye, increased tearing. And then some of the complications of ectropion include inflammation of the conjunctiva, also known as pink eye or conjunctivitis. Also, you can have inflammation of the cornea or keratitis, which can lead to ulcer formations, which can be painful and can lead to scarring, leading to worsening vision and vision loss. But you don't have to worry about this. I'll talk about the management of ectropion later on. So the ectropion scoring system, or the ESS, was developed by plastic surgeons to assess ectropion surgery outcomes and has 8 domains that are scored. Here is an example of a labeled eye that was scored using the ESS, and you can see that there is detachment of the lower eyelid, you can see that there's increased tearing, and you can also see that the lower eyelid has the conjunctiva showing. And we were able to validate the ESS in individuals with EDDs using 6 raters. The inter-rater reliability was excellent, and this was validated using 390 photographs, which you all graciously were able to give us through your participation in the professional photography rooms. So using the ESS, we were able to identify genotypes most affected by ectropion, which were TGM1, followed by ABCA12, followed by SPINK5, and then PNPLA1. And then also using the ESS, we were able to identify the genotypes with the most severe ectropion. So those were ABCA12, PNPLA1, TGM1, and NIPAL4. And then I also told you about earlier some of the scary complications that can result from ectropion, but there are medical treatments for ectropion to prevent those complications. So ectropion can be treated with retinoids, both topical or systemic retinoids. And then these are photographs of an individual with noticeable ectropion who later had improvement, as you can see at the 30-month mark, of her ectropion, and she used topical tazarotene on the lower eyelids. And this was published by Dr. Craiglow, Dr. Millstone, and Dr. Choate. And I want to emphasize that medical management is first line for ectropion, and only after medical management fails should surgery be considered. It's not generally advised to jump straight to surgery because the results of surgery are short-lived, and surgery is an invasive procedure that does have risks. So as you saw, the ESS has many components, and it can be quite cumbersome to use in the fast-paced clinic setting to track ectropion changes over time. So we have created a simpler ectropion scoring tool that's specific for individuals with EDD, but we need your help to validate this new scoring system through your participation, continued participation in our professional photography rooms. Like I said, your participation really matters in being able to validate these tools and employ them in the clinic and research setting and really advance care for individuals with EDDs. Your participation made it possible to validate the ESS, and we're really hoping to validate our EDD-specific scoring tools soon. Thank you so much for your time.
Even if you're already in the registry, please do come for new photos. Now I'm going to introduce Samer Marzouk, who is an MD PhD student at Northwestern, who's going to talk a bit about the work that he's doing in developing this wearable device, along with Sasan as well.
Hello, everyone. My name is Sausan. I'm a medical student at Northwestern.
Hi, everyone. My name is Samer. I'm an MD PhD student, and we'll be talking to you a little bit today about transepidermal water loss, volatile organic compounds, and measuring both of them. Oh, yeah.
Okay. So thank you all for listening to us. But as many of you know, so ichthyosis affects the skin barrier, makes it harder for the skin to retain moisture. Right now, the tools that we do have to measure the humidity that's emitted off of the skin are pretty bulky, mostly limited to specialized clinics and to research settings. And so we have developed this new sensor that is can be worn very comfortably, it's portable, and it also measures this humidity continuously rather than at a single point in time. And so we would love for you to help us kind of validate this new sensor and kind of help us understand what happens to the skin outside of the clinic visit or outside of just a research setting.
Yeah, so I'll just talk a little bit about just the idea of transepidermal flux. And so what the device is measuring is, you can see a picture of it on the screen right now. It's about, not too big, a little bit over the size of under the size of a credit card. And it's a light piece of electronic that we can wrap around with Coban. In the future, hopefully, more consistency with with a wearable band, but for now we're using Coban to adjust to different sizes. And what it does is that we program it to measure the amount of water that your skin is releasing, as well as this other thing called a volatile organic compound. And what that is that your body is always releasing gases, whether it's water gases or non-water based gases. And those gases are usually carbon based. And we're measuring both of them. And what we can figure out from these is some pretty cool things. And so this is from the pilot study from a big paper we did in Nature a couple years ago, where we were actually able to show that in patients with different conditions, looking at ichthyosis as well as eczema, that we can actually differentiate them using the amount of water loss. So in the future, we hope that this can be a tool that patients can use to essentially measure themselves, how much water they're losing, and then we can correlate that measurement to how severe the condition is, and also to bigger things like, is your medication working? How frequently should you be applying the medication? Maybe you should have a higher dose, a lower dose, or maybe you should moisturize more or less. So instead of having to go to your dermatologist and use those clunky lab-based equipment, you can just put this on for hopefully 10 to 20 minutes, working our way down to smaller increments of measurement, and then you can use that in the future. And then a little bit more about the other thing I mentioned. Your skin, beside releasing water, is releasing a volatile organic compound, or a VOC for short. And a VOC is just any carbon-based release form that your body is like releasing as a gas. And this device can also measure VOCs. And right now, that's like a what if, or not what if, a who cares sort of question. But hopefully through measuring more and more, we can figure out what VOCs do, in the sense of we can correlate them with bacteria count, with proteins, and with lipid concentrations. So we can actually be able to correlate these to biomechanical and protein-based mechanisms, and hopefully use that in the future to track disease severity and immune conditions. As mentioned, yeah, so we can measure barrier health, skin function, as well as other conditions more thoroughly and more in a standard way.
So in terms of how, what we can actually learn from your participation at first, right now we have begun, like Samar mentioned, to test this device. We want to continue to test it in a larger group of people with ichthyosis and in people without ichthyosis. So even if you don't have ichthyosis, we still want to get information about how your skin acts and more information about your skin barrier health. And so some of you may have already visited us in our room, so thank you for your participation already. But we are interested in seeing, if it changes, okay. Something that we're also interested in seeing is whether the sensor can detect changes after applying a moisturizer, and that's going to help us understand whether it's responsive to changes happening in the skin in real time. So that's something that we're testing out during this conference as well, and we would love your participation in that. Like I said, just tracking your skin's response in real time. And then, like Samar mentioned, we're trying to really understand more about what these VOCs mean, what can we learn from them, and over time we think that these measurements can really help us track responses to treatments, potentially guide more personalized care. And then ultimately, every person who helps us really helps us build a better understanding of ichthyosis, moving towards kind of having a more specialized, moving us closer to more individualized skincare recommendations and treatment recommendations. So like I said, we are testing this sensor in both individuals with ichthyosis and without ichthyosis. So if you are interested, you can stop by the non-invasive biomarkers room. It would be very helpful for us. And thank you so much again.
Thank you all.
So remember, in order to start doing research, start first at the registration room, which is meeting planner B. Last speaker is Ryland Mortlock, who's an MD PhD student. Ryland is doing spatial transcriptomics work, which is some of the work that's going to be able to show us how to treat ichthyosis in completely new and different ways. He's going to join us now to present his data, and we'll get you to lunch.
Hi, everyone. My name is Ryland Mortlock. I'm an MD PhD student in Dr. Choate's lab. This says, can we use unbiased spatial transcriptomics to understand shared pathogenic mechanisms of ichthyosis? But what this means is, what is one of the things we're using the biopsies that you all have generously donated last time in Albuquerque and this year in Minneapolis? What are we using that for? So one of the ways we're using those biopsies is we're measuring the expression of every gene, all 20,000 genes, in every cell in your skin to try to understand novel therapeutic pathways in addition to the things we already know about what your gene mutation does in ichthyosis. So this is the type of data we produce from each of these skin biopsies. We get this really high resolution picture of the cells, the skin cells that are in your skin, and then we get the expression of every gene in that same tissue sample. We can use that to look at skin cells, immune cells, try to understand inflammation as well as the epidermal barrier. And ultimately, what we're trying to do is build a map of where epidermal differentiation is normally happening in healthy control skin and where it's going off track in different types of ichthyosis, and do this in an unbiased way so that we can come up with new therapeutic approaches that are targeted to certain genotypes of ichthyosis or could be broad across ichthyosis. So I just want to reiterate to say thank you for participating in research, and thanks for having us at your meeting.
So we're so grateful to all of these rising scientists for presenting their work, and Dr. Paller and I and the entire research team are ready to talk with you about how you can be involved and how you can do specific projects along the way. But you know, I think we have three main goals. We want to understand patient experience and disease natural history. We want to expand our understanding of biological mechanisms across all genotypes. And ultimately, we're all looking to develop completely new ways to treat disease. Hopefully, this is just the beginning of what is going to be an incredible period that is going to be a new decade of discovery, which we're going to see a lot more coming from. So thank you so much for your attention.